receptor immediately activates a STAT, and you don't go through this whole long pathway of things that ends up in the activation of the STAT. Okay, so I think that's all I want to say about the JAK-STAT pathway. A lot of people spend lots of time looking at JAK and JAK-STAT pathways. I do want to talk about, are there any questions before we go on? Because I'm going to talk about how do you get rid of those phosphate groups. Okay, so I'm going to leave this on the board. This is activated. Okay, so unlike G protein coupled receptors that just go through a conformational change, these guys go through a covalent modification. So now that the phosphate groups are covalently bound. So that's sort of like a permanent activation unless you have a mechanism to remove the phosphates. So even after the ligand disappears, the phosphates are still there. Okay, so there's really two kinds of proteins that are great at removing the phosphates. Or the receptor. Let's say there's two proteins involved in receptor inactivation. That would be a better. Okay, the easy one is a small, actually a very small molecule. It's on my picture that has two SH2 domains for sure. A little bend and the phosphatase domain. And I'm going to draw it in a different color. Picture or color on the picture. Okay, and the actual catalytic site is here. Okay, this is the phosphatase domain. Okay, these are SH2 domains. SHP1. Okay, so when SHP1 does not have a receptor to bind to, the catalytic domain is covered up, so this is inactive. Because the catalytic domain is inaccessible. But if there happens to be a receptor with some tyrosine kinases on it, okay, they will bind to the receptor, and that swings the catalytic subunit arm so that it is active. Okay, and it can activate, so in this case it's taking the phosphates off of JAK. It can take the phosphates off the receptor. So this is just an example, so it's not limited to removing phosphates just from the JAK. Okay, so SHP1 is active when SH2 domains are occupied. Okay, and I think that's pretty pervasive in cells that you have this kind. You can't use phosphoprotein phosphatases. They tend to recognize a different set or different kinds of phosphate groups.
So just to clarify, the SH2 domains are just binding, and the part that
Yeah.
swung out
So if you wanted to
is
do that
removing.
catalytic domain, it's some other look, or, yeah. So SH2 domains here, but this is the catalytic domain that's actually removing the phosphate off of, in this case, Jack, but it could remove phosphates off of other things, off of the receptor, for example.
Okay.
So you might want to, when you do your cheat sheet, draw the catalytic domain some way else. So that one's pretty simple. Okay, then there's a protein that's even smaller. Notice it is way little. Okay, and I'll just leave this picture on, called SOCS. Okay, so SOCS has one SH2 domain. To dock it to the protein. It's not bound to that phosphate group, it's just in the way. The phosphate group is in the way, okay? I will remove the phosphate so you can see that there's no interaction. Okay. Because it only has, SOCS only has one SH2 domain, so it can only bind to one tyrosine phosphate at the same time. Okay? But it has a docking site. The only other domain on SOCS is one that is going to bind to a ubiquitin ligase. Now, if you wanted to get rid of the signal, what would you ubiquinate? Okay, so this ubiquitination is going to lead to destruction of the protein, not any other signaling. What would you ubiquinate? So if you were this, I don't even know the name of the ubiquitin ligase. Probably the most easy thing to ubiquinate is Jack. So Jack becomes polyubiquinated, not a good thing. Okay, and that leaves Jack to be destroyed in a proteasome. I think if you're going to get rid of the receptor, that's going to involve probably some sort of receptor downregulation, either through endocytosis or autophagy. Okay, so these are the two proteins that are primarily involved in ending the signal created by receptor tyrosine kinases. One's a little more permanent than the other. And I don't think SOCS can work on receptor tyrosine kinases. I think they can only work on those kinds of proteins that have tyrosine kinases that are cytoplasmic. All right. And except for, one other thing, I didn't think we were going to do more today, and I think we'll bring it up. Here it is. Okay, taking a look at, so all we have left actually, which I'm going to do on Monday, is talk about the cardiac hypertrophy experiment. It's that it's a homework assignment that's you're not doing, but it's on Scholar. But I'm going to give you print copies on Monday. And then 15 is the one you do. Okay, but there is one, number 12. It says, what impact does the fusion protein BCR-ABL have on the cell? Where did the fusion protein come from, and how are cancers caused by BCR-ABL fusions treated? Oh, okay. We used to have a coach. I don't know how ago. He's an assistant basketball coach that had the protein BCR-ABL. So chronic myelogenous, I think, i.e. myeloid cells, leukemia. Okay, or CML. Most of them are characterized by having a specific translocation on a chromosome. So I'm going to draw one of the chromosomes. I never memorize what ones they are. Okay, this is chromosome number 9. Okay, on chromosome number 9, it's a gene that codes for the protein ABL, and we'll take a look at ABL in a minute. I'm not going to say much about it. And on chromosomes, so this is chromosome number 9. On chromosome number 20-something, maybe 22, which is a teeny weeny chromosome, is a gene that codes for the protein BCR. And you will see BCR, I'll tell you the domains and you can guess its functions. Okay? A lot of times in cells that have the potential of becoming white blood cells or immune cells, there seems to be a lot of translocation where you get double-stranded DNA cuts and the chromosome, well, you want just regions of a chromosome to rearrange, but sometimes regions between chromosomes exchange. And in this case, you end up with a chromosome translocation which a part of chromosome 22 and 9 swap places. Okay, which one is the Philadelphia? Okay, and this one, this teeny weeny one that has the centromere of chromosome 22, okay, has ABL, and that gets fused to the gene BCR, and this is called the Philadelphia chromosome. And so these two guys together now are actually one gene. It's a fusion gene. It's not two different genes under the same control of of the transcription factors, the same set of transcription factors. This is one gene. So a fusion protein is made, and the fusion protein is called BCR-ABL. Okay, so let me tell you what is the different domains on BCR and ABL. Okay, so we'll do BCR in blue. I'm gonna watch the time, see how much time I have. Okay, it is a serine-reading protein kinase. That's its primary function. Okay. oligomerization domain, meaning, what that means is that BCRs can bind to other BCRs, so they can form dimers with one another, maybe trimers or tetramers, but they will interact or bind to one another. Okay, has a plextrin homology domain, so it could bind to PIP3. A lot of proteins I'm discovering have a plextrin homology domain. A GRB2 domain, binding domain. Okay, in other words, this is a sequence that SH3 recognizes. Okay, so it's a protein kinase that has to form dimers or trimers or something. Okay, that's one for function. Looks like it could be activated by binding to a PIP3, whether or not that domain gets used. Looks like it could be activated by binding to GRB2. I'm not sure which of those domains are actually essential for function. Okay, here's ABL. ABL is a tyrosine kinase. Okay, so now you put these two guys together, they can phosphorylate serines, threonines, or tyrosines. It has a nuclear localization signal. I'm not sure if it's used as a nuclear localization signal. It has an SH2 domain. It has an SH3 domain. So in other words, it could recruit. Oh shoot, I did have a page. I'll tell you what, I'll bring a second page of this BCR ABL on Monday because it'll be good review. It could bind to DNA, it does, and it could have an actin binding domain. Okay, so when you bring together these two proteins into one fusion protein, oops, I'm not sure I can get this full enough. Okay, so this is ABL. Okay, you could get some pretty potent activation of the tyrosine kinase domain of BCR. And that has been shown to recruit a whole lot of typical things. RAS, so the GRB2, SOS, and combined you can activate RAS. Okay, you can get the RAS-MAP kinase pathway going. You can get PI3 kinase recruited. So it can do a lot of different things. Okay, so that the dimerization domain of ABL is really bound to, now stuck to, a BCR, a tyrosine kinase, has turned out to be a lot of a pain in the butt. So there was a drug called, this was the first cancer-specific drug, Gleevec. And I can't remember the other name of Gleevec. That was just the drug company's name for the drug. It begins with an I. Okay, Gleevec is specific for BCR ABL. In other words, it only works in cells that have a Philadelphia chromosome, which is those myeloid cells that are causing chronic myelogenous leukemia. It binds to, blocks the ATP site on BCR. Okay, so that BCR can phosphorylate anything because it won't allow, Gleevec won't allow ATP to bind to the BCR. Okay, so it has absolutely no impact on all of the other tyrosine kinases in the normal cell because it only binds to BCR-ABL fusion proteins. This was, I remember covering this in the biochem seminar because this was so exciting that, you know, you could have some cancer selective drugs, but this was the first cancer specific drug. Okay, so that should do it for a week. So next week, we will do the research. I